All right, cell structure and organization. All right, so again, we're starting to get bigger and bigger in scope here. These are key concepts we'll be going over today. So let's start with microscopy. All right, so I know in the schedule, the part of the textbook that talked about microscopy was not listed. I noticed that. That is a mistake. So there is a section in the textbook about microscopy if you want to go back and read it. Otherwise, I have a beautiful lecture prepared. So, microscopy. We're looking at a few different things when we're looking through a microscope. All right, so how do we know what cells look like? We're looking through a microscope. We're employing microscopy. And microscopy can give us a few different things. All right, so it can give us different resolutions. All right, so the ability to observe two adjacent objects as distinct from one another. All right, so maybe we're looking at chromosomes in a cell. If we have a good enough resolution, we might be able to see two separate chromosomes instead of one blurry blob. All right, we're also looking at contrast. So how different one structure is going to look from another. This can be enhanced by special dyes or special dyes to reveal the cellular structure. So a lot of times dyes will bind to certain types of proteins to allow you to illuminate, say, the nucleus from other types of molecules in the cell. Right? They help you kind of bring out the contrast a little bit better. We also are looking at the magnification. Right? So I'm sure all of you have looked through a microscope or seen a microscopic image before. We're looking at a cell that's super, super tiny, but we can actually see it with our naked eye now. All right, so we're enhancing the magnification, the ratio between the size of an image produced by a microscope and its actual size. So different microscopes are going to differ in how much they will magnify an image. So let's look at the two different types of microscopes. All right, so the type of microscope is going to be based on the source of illumination. Now, the most common one that you've probably seen is a light microscope. All right, we're using light to illuminate and enhance something that we're looking at under the scope. It normally has a resolution of 0.2 microns, so we can see pretty tiny under that microscope. But the other type of microscope that you probably have not seen, and if you have, that's awesome, because I haven't even seen one of these in person. Electron microscope. All right, so an electron microscope, it doesn't use light anymore. Instead, it uses a beam of electrons to help illuminate what you're looking at. All right, so I don't expect you to go into any more depth on that for electron microscopes. They're awesome tools, though, because they have a really, really high resolution. All right, we can see even smaller with an electron microscope, down to 2 nanometers, which is 100 times better than a light microscope. They're pretty cool. It's basically based on the principle that the wavelength of this electron beam is much shorter than the visible light wavelength, so it can capture more intricate details than what a light wavelength could do. All right, so if we're talking about light microscopy here, we can go in even further and talk about different types of light microscopy. All right, so the ones that we're going to be using in labs, the ones that are going to be most common because they're economical, is the standard microscope. Right, we're using the bright field microscope. So this is where light is going to be focused through a glass lens. It's going to pass directly through a sample. All right, there's nothing too unusual about it. We also have these phase contrast microscopes. Right, so they're really good at amplifying differences in the different phase of light transmitted or reflected back by samples. So now we're starting to sort out different wavelengths within the light to get different contrast within the cell. All right, so we're improving the contrast of a lot of denser structures. And then we also have this type of microscopy called differential interference contrast, all right, or DIC, also known as Nomarski microscopy. All right, so it's another method that we're using light, reflecting light back to us in different ways to help improve the contrast, and it's really good for looking at internal cellular structures. All right, so what does this mean visually? All right, So these are the different types of microscopy when we're talking about a light microscope. All right, we can see this one here. This is the standard light microscopy. We're just passing light through a sample. All right, it doesn't seem to, we can't see anything too distinct. All right, the resolution is there, but the contrast is not. Versus this phase contrast microscopy. All right, now we're starting to see a little bit more detail within the cell. All right, we can see these darker spots are the denser images. We're starting to see more contrast. We can see the nucleus more clearly. And then the same with this differential interference contrast. All right, we're seeing more contrast than just with the standard light microscopy. All right, we can clearly see the nucleus. It's coming back to us as a slightly different image than the phase contrast, just based on the ways that we're manipulating the light. We also have fluorescence light microscopy. So you might have seen these type of images in a biology setting before. All right, so this is where we're basically putting in a fluorescent molecule and having that bind to specific proteins to help improve the contrast that we're seeing. All right, I'm not going to go into this too in depth, but I wanted to throw this out here that there's another type of microscopy. We're using these fluorescent molecules. And depending on how you use it, what type of microscope you're using, you can get more or less a better resolution on it, better contrast. All right, we also have electron microscope types. All right, electron microscopy is awesome because we can see really, really small images. But we have two basic types, the transmission electron microscopy and the scanning electron microscopy. So in electron microscopy, we're shooting a beam of electrons at our sample. All right. In transmission electron microscopy, we're shooting that beam of electrons through the sample. All right. It's passing right through that sample. These samples are normally laced with heavy metals. All right. And those heavy metals are going to deflect electrons back and help give us this image that we're looking at. All right. So some electrons are going to be scattered, while others are just going to pass through, and that creates our highly detailed image. All right, they're normally cross-sectional. We have a very good image, but electron microscopy is very expensive, and we cannot do this on a living cell. All right, so the cell is going to be dead at this point. Now, scanning electron microscopy uses the same basic principles. All right, we're shooting a beam of electrons at the sample, but this time the whole sample is coated in this metal covering. All right, so it's coated in this heavy metal covering. It's going to scan the surface and make a 3D image. All right, so we're not looking at a cross-section anymore. We're looking at the structure as a whole, looking at the surface. It's usually used on non-living cells. All right, I think you can use this on a living cell to some extent, but it's mostly on non-living cells. All right, that was a lot of talk on microscopes. All right, I know it's a little dry. So I'm going to have you guys group together and talk amongst yourselves to answer these few questions.
Sure. Yeah. Yep.
All right. Good.
Yeah. Appreciate it. Thank you.
Oh, yeah.
And then you can get like a banana.
All right, looks like most of you have these answered, so let's come back together and discuss. All right, so I want to observe a cell, and I want to be able to see all the different structures inside of a cell. All right, so what type of microscope could I use? Yeah?
We have the differential interference contrast microscope because you can see like the nucleus like that, and you see a little bit more of the 3D structures.
Yeah, totally. And a little bit more broader, what type of microscopy is that? Light microscopy. Yeah. Did anybody have anything different? Yeah? Okay, we could use phase contrast, still in the light microscopy zone here. Now I'm going to say you could also use electron microscopy for this. All right. It's going to be even more fine detailed, but both of them are going to work. What type of microscope would allow me to see the smallest of details? Electron. Yeah, exactly. Kind of just answered that for you. What type of light microscopy would give me a good contrast between the different structures? We can use fluorescent microscopy. Some of the other ideas that were shouted out earlier, we could use the DIC, we could use the phase contrast. They're going to give us more of a contrast in our cell than just a basic light microscopy. Does the type of microscope I choose matter if I want to observe a living cell? Yes, all right. Am I going to choose an electron microscope?
No.
No, all right. You guys seem to have a pretty good grasp on this. Any questions?
No.
All right, moving on. All right, so the overview of cell structure. All right, so we kind of learned how we're going to look at a cell. Now let's actually look at them. So we have two categories of life, two broad categories of life that we can separate our cells into. Prokaryotes and eukaryotes. All right, so prokaryotes, they're going to have a very simple cell structure. They're not going to have a nucleus. All right, they have nucleoid regions. All right, so regions that are going to contain their DNA, but they're not membrane bound. They're not bound inside of a nucleus. Now this is directly contrast to a eukaryote. All right, eukaryotes are going to have way more complex cells. All right, they're going to have a lot of organelles. They're going to have DNA enclosed within a membrane-bound nucleus, not just free-floating. Many, many different types of organelles. Anything else I wanted to say about that? We'll talk about organelles a little bit more specifically next week, but they're just membrane-bound structures that have very specific functions and structures to them. I put this table in here so you can kind of directly contrast the difference between prokaryotes and eukaryotes. All right, so the location of the DNA is going to be different. A nucleoid, which is just a region where the DNA is contained, versus a nucleus. Internal membranes and organelles. Eukaryotes are going to have way more complex organelles, a wide variety of them. The cytoskeleton, which we'll get to in just a little bit here, is very limited in prokaryotes, but very extensive in eukaryotes. And the overall size, I'm just going to flip back to that slide. You can see a prokaryote, very, very tiny compared to a eukaryote. All right, so within prokaryotes, we have two categories of prokaryotes, bacteria and archaea. So both of them are going to be very small. All right, we're talking 1 to 10 microns in diameter. Bacteria, they're going to be very abundant, and they're very abundant in the environment as well as in our bodies. They can be found in soil, water, digestive tracts. They play very vital roles. I know within the past couple of years, there's been some cool studies out there about how the bacteria in our gut can influence our mental health. Right? So there's a lot of fascinating things going on with bacteria. Yes?
Is this the one that termites have to digest the wood? Correct?
Yes, I believe so. I think you looked ahead, didn't you?
Yeah.
The video?
Yeah.
Yes. So yes, I believe that is a bacteria that helps them do that. The vast majority, they're not going to be harmful to humans. In fact, they're pretty beneficial to us. Some species, though, can cause diseases. All right, so the archaea, on the other hand, very similar in size, very less common. So they're often found in these very extreme environments, such as deep sea vents, some hot springs, petroleum deposits, or really extremely salty waters. All right, so if you think of an extreme environment, you're probably going to be able to find archaea living there. Now for this lecture and this semester here, we're only going to focus really on bacteria, mostly on eukaryotes, but that's all I'm going to say about archaea. All right, the biggest thing about these two prokaryotic cells is that they lack internal compartmentalization. All right, it really sets them apart from those eukaryotes. So let's kind of look at the structures of a bacterial cell. All right, so a bacterial cell, the first thing we kind of want to point out, it's kind of our guiding theme here, is this plasma membrane. All right, so the plasma membrane, it's a phospholipid bilayer, which we learned about when we talked about fats and carbohydrates here. That lecture 2-3 times ago, we talked about them. And we will be continued talking about them more in depth. But this is our plasma membrane. It contains those phospholipid bilayers as well as embedded proteins within that. And it basically is going to form a barrier between the internal cellular environment and the external cellular environment. All right, so if we look at the environment inside of our cell, inside of our plasma membrane, there's a few things to note. All right, the first is the cytoplasm. All right, so that's basically everything inside of the plasma membrane is the cytoplasm. All right, this is the cytometabolism happens within here, contains everything inside. We also have the nucleoid region. All right, so this is where the DNA is more or less contained. It's not in a membrane-bound envelope. All right, it's just free-floating, but kind of all stuck together there. And then we also have our ribosomes, kind of free-floating here. All right, so ribosomes, very important in polypeptide synthesis or the formation of proteins. All right, so let's look outside of the plasma membrane now. All right, so just to reorient ourselves, the purple plasma membrane here. Outside is this kind of deep gold-colored layer. All right, this is the cell wall. All right, so the cell wall, it's rigid, it provides support, it provides protection, but even though it's rigid and helps protect the cell, it's still very porous, so it can allow a lot of nutrients to reach that plasma membrane and to go inside of the cell to help support all of its functions. All right, now outside of the cell wall, we have this glycocalyx, all right, this really thick yellow part here. All right, this is a very viscous covering. And it traps water in to help prevent desiccation of the cell, helps prevent the cell from drying out. But it can also help to give protection because it is so thick. All right, because it's thick, it's going to help it, say, pass through the digestive system of an animal without getting harmed. All right, a couple other things sticking out of this glycocalyx. We have the pili. All right, there are these hair-like projections. They're used for attachment of the bacteria cells to each other or maybe to another structure inside of our bodies. And then we have these flagellum. All right, so they're very long appendages, and they're going to be used for movement. All right, so compared to eukaryotes, very simple, not a lot to kind of remember about these. But any questions? All right, so let's move on to eukaryotic cells. All right, the biggest thing about these cells is DNA is going to be housed inside a membrane-bound nucleus. All right, and everything inside of these cells have very compartmentalized functions. All right, so we have organelles, which we kind of mentioned before, membrane-bound compartments. They each have a unique structure, and because of their unique structure, they have a unique function. All right, it's that theme we're beginning to see emerge in this class: structure equals function. There's also a large variety. All right, so the shape, the size, organization, they're all going to vary considerably. Depending on if you take a cell from a plant versus an animal, you're going to see a lot of variety. Even if you take a cell from, say, your brain versus your gut, all right, you're going to see a lot of differences between those two cells. All right, there's a lot of structures in eukaryotes. We're not going to go over them now. This is what we're going to talk about on Monday. But there's a lot going on here compared to the bacteria. All right, so this is an animal cell. That's a plant cell. They're different, but they have a lot going on and a lot of similarities. All right, so what's going to determine what a cell has inside? All right, so how does a single organism produce different types of cells? So in this image here, we have a skin cell versus a neuron. All right, two very different types of cells. All right, so how do we get those cells? All right, so all of our cells, they have identical DNA. All right, we have the same DNA in every single cell that we have in our bodies. But they have different proteomes. All right, so a proteome is the complete protein composition of a cell or an organism. All right, so proteome is describing the proteins you have within each cell, basically. All right, so we know structure equals function. So depending on what proteins we have is going to depend on how this cell is going to function. And why these protein profiles differ is something called differential gene regulation. All right, so every cell is going to express a unique set of mRNAs, which in turn will be translated to proteins. And we're going to talk more in depth about that process later. All right, but know that we have the same DNA, but the amount of proteins expressed might be different. The levels of expression, so maybe one cell has 100 times more of this specific protein than another cell. They still have the same proteins, but they're expressed in different amounts. Which subtypes of proteins are expressed? All right, so just different proteins that have slight molecular changes could be expressed differently in each cell. And then post-translational modifications, which is basically saying maybe that protein has folded slightly different from another protein. All right, now that's way beyond the scope of this course. All right, I just threw that up there to show you that there's more ways that you can change the different proteomes between different cells. This can be very relevant to diseases. All right, so healthy cells have very different proteomes to those of cancerous cells. All right, the way that the proteins are expressed. Maybe they're overexpressed, underexpressed, whatever it is, it's leading to a cancerous type of cell. All right, so as a group, think about what are the main differences between our two different cell types, but what are some of the similarities? All right, have at it. Oh, yes. Where's the I know there's one like down the hallway a little bit before I get in the hall. Okay.
I know, I'm actually closing, I can't even hear myself. Okay. Correct. Yeah, I have sinus. Sinus are way smaller, but like I said. I'm going to bathroom. I'll be right back. I can't listen to myself sniffle.
All right, let's come back together and discuss here. So, who could tell me what one of the main differences is between prokaryotic and eukaryotic cells? Yeah?
The nucleus isn't found in prokaryotic cells, it's more floating around in cytoplasm as compared to eukaryotic cells.
Yeah, a nucleus versus a nucleoid region, definitely. What's another difference? Yeah? Differences in size. Yeah, eukaryotes much larger.
Eukaryotes have a bunch of different kind of organs or smaller.
Yeah, so eukaryotes have a lot of different organelles in them.
The difference in the cytoskeleton, the prokaryotes being limited, the other one.
Exactly, prokaryotes, very limited cytoskeleton. Any other differences we want to mention? I think you guys touched on all those main differences. What are some of the similarities, though?
Their cell.
They're both cells, yes.
They both have cytoplasm and ribosomes.
They both have cytoplasm and ribosomes.
They both have DNA.
They both have DNA, yes. Any other similarities?
They both have like some kind of membrane to encompass everything.
Yeah, so they both have a plasma membrane. Yeah. Anything else?
They can both be
So they both have a type of cytoskeleton inside of them. Yeah, yeah. All right, awesome. Well, speaking of the cytoskeleton and the cytosol, let's move on into that direction. So, the cytosol, that's all the intracellular fluid that's inside of the plasma membrane. All right, so it's regions of the eukaryotic cell that is outside of the cell organelles, but inside of the plasma membrane. All right, it's that intracellular fluid. Now, cytoplasm, all right, includes everything inside of the plasma membrane. All right, so the cytocell is just the fluid, but the cytoplasm encompasses the cytocell, the endomembrane system, so the membranes of all the organelles inside of the plasma membrane, and all these semi-autonomous organelles. So those we'll talk about next Monday here, but those would be mitochondria or chloroplasts. All right, so basically they grow and divide independently, but they still rely on the cell for some of its components. All right, so cytocell versus cytoplasm. All right, so the cytosol is going to be the site of the main coordinating region of the cell for metabolism. All right, so metabolism, some of all those chemical pathways that are either going to produce or utilize energy. All right, and there's a couple different main metabolic pathways that we'll talk about briefly here. All right, but catabolism is basically a breakdown of a molecule into smaller components. All right, so maybe we need to utilize stored energy. We need to break those large polymers down into units that we can use for energy. It might be breaking down components so we can use them as building blocks for other molecules. All right, so that's in contrast of anabolism, which is essentially the opposite. This is where we're synthesizing cellular molecules and macromolecules, right, such as polysaccharides or proteins. All right, and again, the cytosol, this is the area of the cell that is the main coordinating region for all these metabolic activities. All right, so a lot of times we think of this cytosol as kind of an empty space, all right, but there's actually a lot going on in it, all right, and mainly in the cytoskeleton. All right, so the cytoskeleton is what provides the cell with the shape, organization, and movement. All right, so basically we have a network of these three types of protein filaments that all help to provide shape and movement inside of the cell. So we have microtubules, which are long and hollow. They are made out of tubulin proteins, and they have a polar structure. All right, now they also show dynamic instability, which means they can oscillate between growing or shortening. All right, so they have a growing phase, they have a shortening phase, they can oscillate between them. All right, so this might be important when we start talking about cell division. All right, this plays a key role in cell division. Being able to lengthen and shorten those microtubules helps line up the chromosomes in cell division. Now we also have these intermediate filaments. All right, they're intermediate in size, as the name might suggest, and they form twisted, almost like rope-like structures, and they're relatively permanent. All right, they really don't shorten or lengthen. These really help maintain cell shape and rigidity. All right, so they're very more or less stable. And then we also have these actin filaments. All right, they're going to be very thin. They're going to be very long. They can also be called microfilaments. They are also polar. All right, so microtubules and actin, they are the polar filaments here. They're highly concentrated near the plasma membrane, as they're often anchored into membrane proteins. And they really help, again, aid in cell shape and strength. All right, so here's an image of all three that we just talked about. So you can kind of visualize. All right, so the microtubules, they're hollow, they're thick, they're polar, they have a plus end, the negative end, they're made out of tubulin. All right, we can kind of see them highlighted in this fluorescent microscopy down below. These intermediate filaments, very rope-like, they're twisted about. They are not polar. They don't have a plus and a minus end. And then we have our actin filaments. All right, they're very long, they're very skinny, they are polar, and they are made out of an actin protein. All right, I think these images do really well in showing you just how much of this is in that space of the cell that is often thought of as empty. All right, so motor proteins are also crucial to the cytoskeleton. All right, so motor proteins are going to help things move around, whether that's organelles, whether that's any type of cargo they need to move. Now there's three domains to a motor protein that are going to help it move. All right, we have the head, the hinge, and the tail. All right, so in this image here, we have our head, we have our hinge, and we have our tail. Right? And this aids in three kinds of movements that we're going to talk about. All right, so we have motor proteins that are going to carry cargo along the filament. All right, maybe we need to move some building blocks around to get to another part of the cell. We also have it where these motor proteins, they're going to stay in place, but the filament is going to move from under them. And then we also have a type where both the motor protein and the filament, they're both stable, they're not going to move, but the action of the motor protein is going to cause a microtubule to bend. All right, we're going to go a little bit more in depth in all of these. And this is where we sometimes get what's called the walking analogy. All right, where the ground might be the cytoskeleton, your leg is the head of the motor protein, and your hip is the hinge. All right, and I'm going to throw some visuals up because I know this is a very visual thing to learn. Before I do that, let's just kind of go over one process. All right, so again, we have the head, we have the hinge, we have the tail. All right, now we use ATP to aid our movement here. And ATP is going to help with the binding of the head to the filament. All right, binding and release of the head. So in here we have an actin filament. All right, we have the head that is bound onto it. First thing that's going to happen is that head is going to release. It's then going to bend forward and attach to the actin further along that actin filament. All right, and then it's going to bend back and basically move the filament from underneath of it. All right, so it's moving that filament underneath of it kind of by this bending motion of the motor protein itself. All right, let's take a look at some videos here to help demonstrate this a little bit more. I just want to show the very intro of this video so you can kind of see that movement taking place. All right, this is a very long video. You can watch it all if you wish. I don't expect you to at all.
Hello, I'm Ron Vale.
And we
in
don't
this
have
talk,
audio.
I'd like
All right.
to introduce you to molecular proteins.
All right, well, we don't need audio for this video. This is just basically showing how that motor protein is walking along.
These proteins are able to walk along a cytoskeletal
All right,
tract and
looks like
transport
something's
a
walking
variety of
along
different
a microtubule.
inside of cells. What I'd like to do today
That is all I wanted to show from that video because I think that gave a great visualization. Now, another video I wanted to show, I actually do need audio. Is anybody really good with technology here?
Can you turn the closed caption?
I definitely can, yes.
I don't know if they're on there already. But, you know.
Does anybody know how to get the audio throughout the room?
Are you able to just like could you just play it off your computer? Yeah.
I can try and see how loud we're going to go.
Our brain is made of billions of nerve cells, and they're all connected. If we take a closer look, a nerve cell seems to have antennas. Most of them are receivers of information, but only one is a transmitter, called the axon. This axon is connected to several receivers of other cells, forming a gigantic neural network, the brain. Meet John. John is a kinesin, a motor protein. He lives inside a nerve cell, and he has a proper job. To ensure that a brain cell does his job properly, it needs the continuous flow of building materials, proteins. They travel through the cell using the cytoskeleton. If you would compare a nerve cell with a city, the cytoskeleton inside the cell would be the roads, and the traveling proteins would be the traffic. These materials are towed by motors along the roads. And just as in real life, there are different kinds of motors and different kinds of roads. John's sole purpose in life is to deliver his cargo to a specific place in the axon. He takes the main roads, and he walks in just one direction only. John's job may seem easy, but it's not. He has to overcome a number of obstacles to ensure that the right amount of cargo arrives at the right place To make the journey even more difficult, John is not alone. Other motor proteins ride along with his cargo. They haven't woken up yet, but that will happen soon. The journey starts in the center of the city, just like in the center of the cell. To enter the axon, John has to pass a place called the axon initial segment. In this segment, there are two kinds of roads. The main roads that John uses, called the microtubules, and a lot of little alleys called actin. And here, our brave motor protein meets his first challenge, because one of his sleeping travel companions, myosin, has woken up and starts to cling to the actin. And there are a lot of alleys. Only brute force can save John now. Fate strikes again. The other companion, Dynin, wakes up, and he can only walk in the opposite direction of John, resulting in a tug of war. But there can be only one. Along the axon in which John travels, there are places called synapses. Here, the axon connects to receivers of the other cells. Regulating proteins call the shots here. This traffic police makes sure that all passing traffic gets to the right destination. If John's cargo is needed in this synapse, he will be stopped and myosins take over his load. But today, John's cargo is safe. But what he does not know, that his road is under construction just a few blocks away. In our nerve cells, the cytoskeleton is changing constantly. Roads are built, but are also broken down. Facing this kind of obstruction, John has to find a detour. John isn't the only motor protein on the road. There are many more. Our nerve cells need a smooth traffic flow in order to perform well. A traffic jam due to problems during the journey may ultimately result in brain disease. Understanding the challenges John faces could improve treatment or prevention. Finally, John arrives at his destination. He has fulfilled his destiny. But several other Johns are just getting started.
Alright, so I thought that was kind of a cute little animation that demonstrates a great analogy. Yes?
Okay, why do things that work against you exist?
That's a good question. And I'm assuming it has to do with the takeover at different parts of the cell. So like they talked about, if you had to go through a synapse, maybe that myosin would wake up and take over for him.
Okay, gotcha.
We're going to talk a little bit about dynosin. Now the complexities about them existing on the same cargo, it's not really my realm, and I don't have a good answer for that. That would be kind of what I would guess, is so it can take over and do some other function. All right. So I know I covered a lot about like neurons and axons, which you can kind of ignore for the most part, but I think the analogies in there are great, and I love his little slippers. So I thought this was a fun little video. All right. So let's go back and kind of talk about these a little bit more here. All right, so the analogy of John that we just saw here was an example of a motor protein moving. All right, so the microtubules, they're staying in place, but the motor protein is moving along them. All right, so we have the cargo, and we have our motor protein moving along. All right, so another way that we can have movement inside of our cell is what we had talked about previously, all right? So instead of the motor protein moving, all right, we have the actin filament moving, all right? So the motor protein stays in place, and through the movement of bringing its head up and further along the actin and back down, pushes it below it. Now we also have a third type of movement. All right, this is where both the motor proteins and most of the microtubules are stationary. They don't move at all. All right, so they're stationary, but part of the motor protein, specifically dynin, all right, starts to move and moves along another microtubule. But because they're both stationary, but part of it's moving, it's causing this bend to occur in the microtubule. All right, so this is very common in flagella and cilia. All right, so flagella, they're normally larger than the cilia. They're present generally singularly in pairs. It helps aid in movement, so think of a sperm cell. Generally present in a 9+2 microtubule array, which I will explain in just a moment here. Cilia, they tend to be shorter than the flagella. They tend to cover kind of the entire, or at least most of the cell surface. All right, but movement in both of these is because of a propagation of a bend. All right, beginning at the base and moving towards a tip. Now, I guess I had one more thing to show there. But just to show you, I mean, this is a flagella. They're very long. This is a sperm cell. It's existing just as one. Now, they can exist in clumps, like that bacteria example we saw earlier. And then we have these cilia. All right, many cilia along the entire surface of the cell. Now, I'm going to stop there because we are out of time. When we come back next week, I only have a couple slides left and another cute little video to help demonstrate this exact movement of a bend, because it can be a little harder to wrap your head around. But there is some prep for Monday. All right, make sure you complete the quiz before tonight. Watch this video before Monday. And then I am assigning you guys an assignment. All right, it's not due until the 27th, so you have plenty of time to complete it. I would complete it well ahead of the exam, though, so you're not having competing things due. But it's basically a popular science article assignment. I want you to find an article. I want you to tell me, what's the summary of it and how does it relate to the course material, right? Now, what I uploaded does not say this exactly, but it's with 8 points, and it's due midnight, September 27th. I will change that on the Moodle site as soon as I get a chance. All right. Yeah, you're welcome.